Hello and welcome to our lecture dedicated to the cell fate. Today we are going to be learning how the cells decide when to live, when to die, and when to generate specialized cell types. As usual, I'm going to briefly recapitulate what we have learned in our last cell biology lecture dedicated to cell signaling. We have learned that all of our cells must adapt to changing environmental conditions. Our cells need to change gene expression patterns in response to injury, disease, pharmacological treatment, but also to other things, for example, like changing of temperature. We have also learned that most cell communication involves exchange of biochemical signals, and also that multicellular organisms are also able to sense the presence of other cells adjacent to them. Those signals or stimuli can include growth factors, cell-to-cell contacts, hormones, touch or temperature. Lastly, we have learned that signal transduction is the mechanism by which the cell is able to respond to the stimuli and change the gene expression pattern. The first type of cellular communication we had a look at are quite local communication methods, and the first type of this local communication are cell junctions. Here, neighboring cells are able to communicate with the neighbors by direct exchange of information. In addition to this, cells can communicate by interaction between molecules on their surface, where one cell is actually acting as a signal-giving cell, and other cell can sense the signal by using specialized receptors, activating thereby different signal pathway transduction pathways. Cells can also communicate via secretion of molecules, and the first type of this type of communication we had a closer look at is paracrine signaling. Here, signal molecules are produced and secreted by the transmitting cells, and then those signals diffuse locally, bind to receptors on the target cell, and activate different signaling pathways. A very special type of this paracrine signaling is synaptic signaling, which is also quite local. Here, neurons secrete so-called neurotransmitters, the signals. Those neurotransmitters diffuse across the synapse and bind to another neuron, which is then stimulated. Over long distance, our cells usually communicate using the so-called endocrine signaling, and this signaling is mainly mediated by hormones. In endocrine signaling, hormones are released by one cell type and received by another cell, which can be quite far away from the sending cell. Those hormones are made in one part of the body and then transported via the bloodstream to the receiving cell. The recognition of the signals is a job of a very specialized type of proteins on the surface of the cells called receptors, and the signals are referred to as ligands. Like enzyme-substrate interaction, a ligand fits its receptor like a key in the lock. Cell signaling per se can be divided into three phases, and the first phase is reception, where the ligand binds to the receptor. Second phase is the transduction, where a signal is usually transduced is transduced within the cell, and this usually occurs with amplification of the signal. The last step is the response, where certain cellular processes are activated, and those processes can include any random process in the cell you can imagine. So this response could be activation of migration, proliferation, cell growth, or cell death. As I mentioned before, signal transduction usually involves amplification. This amplification results in a much stronger biological response and is usually achieved by several enzymes in a chain. So, What are we gonna be learning in the session today? After watching those videos and after a bit of revision, you should be able to describe the difference between cell growth, cell death, and cell proliferation. In addition, you must be familiar with the cell cycle and its restriction points. So those restriction points are also very, very crucial in cancer cells. You also should be able to understand the basics of cell type specification or differentiation in multicellular organisms. You should be able to demonstrate an understanding of the importance of cell signaling and regulation of gene expression for differentiation. Moreover, you should appreciate how cells in multicellular organisms regulate their fate in terms of signaling. Last but not least, you must know how deregulation of common cell fate mechanisms contributes to cancer development. So, what is cell fate? Cell fate can be quite various. So, a cell can survive. A cell can also die. Moreover, we can have division or proliferation. We can also have differentiation, and this is actually quite common in stem cells because stem cells are uncommitted cells. However, if injury or disease occurs, those cells can reactivate the cell cycle, proliferate, and then specialize or differentiate into very, very different cell types, including, for example, nerve cells, blood cells, muscle cells, and so on. So, as we have learned last week, our cells are constantly exposed to many signals and they must be able to adapt in response to those signals. And the first and most common signal a cell could receive are signals leading to cell survival. So what you need to keep in mind here is that if cells don't receive survival signals, they usually undergo cell death. In addition, if a different set of signals is received by the cells, this could lead to activation of cell proliferation or cell division. Moreover, cells could differentiate if a different set of signals is received, and lastly, the cells could die. As you can see already on this slide, the basic set of signals cells are receiving are actually survival signals because they are required for not only for cell survival, they are required for cell division and differentiation as well. So those signals are here represented by the letters A, B, and C. If a different set of signals is sensed by the cell, represented by the letters D and E, they divide. If another signals are present, represented by the letters F and G, those cells are gonna be differentiating. So again, A and G are very, very distinct signals mediated by different receptors that are stimulated by different type of ligands. So different combination of the signals lead to the cell undergoing very, very different cell fate. So first, let us have a look how the cells decide if to survive or die. Survival signals are here the key because cells require signals continually from surrounding cells to survive. If those signals are not present and death signals are received, this leads to cell death, and this cell death is usually very, very controlled, but we will have a dedicated lecture on cell death in next term. So, what signaling pathways are in charge of cell survival? The most important survival pathway you should keep in mind is the so-called IGF-1 signal transduction pathway. IGF-1 stands for insulin-like growth factor 1. The signal transduction cascade involved in cell survival is activated by IGF-1, which is the ligand. IGF-1 binds to the respective receptors, which leads to recruitment of several adapter proteins and activation of downstream kinases. Those kinases produce second messenger and activate downstream proteins, including RAS. And RAS is quite important because this protein is key in the development of cancers. But we are going to be talking about RAS later on as well, and also during a few lectures in part 3, where I'm going to be talking about personalized medicine, which is currently developed to tackle cancer. So, IGF-1 signaling pathway inactivates death protein and promotes cell survival. And this is how survival is achieved. If death signals are present and survival signals are lacking, cells undergo programmed cell death. And what I would like to show you now is how this actually looks in a dish. Each of those round circles here represents one single cell. In this experiment, death signals have been artificially introduced into the culture, and if you look at those cells, after receiving those signals, the membrane undergoes quite dramatic changes, and if you look at this cell, then you will see that fragmentation of the membrane occurs. A lot of cellular structures disintegrate. However, this process is quite controlled because it results in production of vesicles, and all of the former components of the cells are embedded within those vesicles, thereby limiting inflammation in the tissue. So, how does apoptosis or programmed cell death work? First, the cell shrinks and the chromatin condenses. Later on, the membrane starts blebbing and the organelles disintegrate. Afterwards, the nucleus and organelles collapse and membrane continues to bleb. Then apoptotic bodies form, and they look sort of like vesicles you've seen on the video I showed you a few seconds before. This is quite important because those vesicles or apoptotic bodies can be digested by macrophages in the human body. Therefore, no inflammation takes place because all the cellular compartments are tightly contained. So, if death signals are received by the cell, apoptosis can take place fairly quickly. This is because our cells are always ready to respond to death signals. Apoptosis is also fairly quick because no new gene activity or protein synthesis is required. This mechanism is highly controlled and organized, unlike other types of cell death, for example necrosis. But we are going to be looking at necrosis in our lecture dedicated to cell death. Death signals are mediated by specific extracellular ligands that bind to specific cell surface receptors and initiate intracellular signals. And those signals are usually tumor necrosis factor, Fas ligand, trail, and trail. Those signals bind to the receptors. This leads to recruitment of adapter proteins and finally activation of different cell death cascades. But again, we are going to be looking at cell death in detail in our dedicated lecture in the spring term. With this, I would like to end our first session of this lecture series, and I will see you in session number 2. Thank you very much.